Good morning, everybody. I know that it takes just a few seconds for people to start joining us this morning. We do have a few updates we want to start getting into right now. We, of course, have a really great presentation I think a lot of people are excited to hear about. But before that, we want to start off by honoring one of our community members, Dr. Steve Coutre. I think many of us knew pretty well. I knew him quite well through my training and throughout the years I've been here. Dr. Coutre was a key member of our community, and I think many of us heard recently about his passing. We have Dr. Ravi Majeti, of course, Division Chief, who's going to say a few words to honor him. So thank you, Dr. Majeti, for being with us here today to say a few words about Dr. Coutre.
Thank you so much, Errol, and good morning to everybody. It's really an honor and, of course, sad to highlight the strong memory of Dr. Steve Coutre. I'm sure many of you know Steve, worked with Steve, and his contributions to Stanford medicine and Stanford hematology in particular are numerous. You may know that Steve came to Stanford as a fellow and in the mid-1990s joined the faculty to really reinvigorate our clinical hematology practice and research program, and really establishing Med8 as a leading service for the care of patients with heme malignancies. Steve also established an internationally recognized clinical research program across a number of diseases, but made this most of his impact in the disease CLL, chronic lymphocytic leukemia. He participated in a wide range of clinical research activities. You may know there are NCI-funded cancer cooperative clinical trial groups, one, the Southwest Oncology Group. Steve served on the leukemia committee and was one of the co-leaders driving many of those initiatives forward. At Stanford, he developed and led our cancer clinical program, our clinical research group, and our outpatient practice for many years. And needless to say, he mentored countless fellows and faculty, myself included. On a personal note, when I became chief a few years ago, I turned to Steve to say, hey, Steve, how do you feel about having a chief who was your fellow and that you trained? And he was so supportive, so enthusiastic, and having that for me personally was really a big impact on me stepping into this role. Stanford hematology is deeply indebted to Steve. He will be deeply missed, and our condolences to his family and, of course, his friends. And these pictures here just highlight Steve, his many activities right up in this last year, CLL and COVID, educating the community, educating colleagues, giving plenary presentations at scientific and clinical meetings, just a giant, and he'll be very much missed. We have set up a memorial fund in Steve's honor to support education and research in the division. His passions and any contributions will be greatly appreciated. Thank you so much, Errol.
Thank you, Dr. Majeti. And when we lose community members like this, it feels like there's no great way to fully honor how amazing and what an impact he had in our community. So we'll continue to do so in any way possible. If anybody has anything they'd like us to do, please let us know. Of course, we sent out some communications last week, but it feels like there's no full way to fully honor him because he was such an important member of our community. Thank you for being with us today to say those few words. We'll make that tough transition over now to our grand rounds. But again, if anybody wants to help us in any way honor him, we are fully available to do so. So thank you again. We're going to move to our usual updates. Thank you, Dr. Collins, for being with us as we continue to progress through the pandemic and COVID. We wanted to occasionally update you on numbers throughout the community. So Dr. Collins is here to do that.
Thanks, Errol. So I'll just quickly run through some numbers. Starting with the curve we've seen often now of our 7 Bay Area counties in terms of hospitalization numbers. You can see that we are certainly down from our peak for this Delta wave, although interestingly, there was this little sort of peak on the way down, which if you actually look, it looks like that was in Santa Clara County if it's broken out by the different counties, although fortunately seems to have come down. As far as cases in the Bay Area, I just went and looked at the different counties surrounding our hospitals. You can see for Santa Clara and Alameda here, they're broken down by vaccination or not vaccinated. San Mateo is a gross number of cases. In general, cases are down, although interestingly, in the unvaccinated in Santa Clara, there has been a rise in the recent weeks. As far as Stanford, our positive cases and our hospitalized cases by day have consistently come down, and we've kind of been at sort of a steady level Our census has been around the same for the last several weeks, even down a bit recently, as far as COVID patients, I should say. As far as demographics, we identified now 467 adult hospitalizations within 14 days of a positive RT-PCR. That's since July 1st. You can see the breakdown by our hospitals. Still a little bit more male than female. Most represented race and ethnic groups are white and Latinx, and the age distribution is on the bottom right there. Something interesting, just looking at gross numbers in terms of those needing ICU care with a positive test and mortality rates. It does appear that for the Delta wave, our ICU and mortality rates were higher. If you compare it to March 2020 to March 2021, interestingly, our readmission rate was about the same. And then very quickly, thought we would just mention that we had looked at pediatric data in our hospital systems. We did identify 65 hospitalizations of pediatric patients within 14 days of a positive test since July 1st, about equal male to female. When we looked at the race ethnic backgrounds, interestingly, those of Latinx background were pretty overrepresented at over 55%. And then you can see the age ranges there at the bottom. So overall, hospitalization and cases are down. Hopefully they'll stay that way. Interestingly, we did see higher ICU and mortality rates during the Delta wave. So thanks.
Thanks so much, Dr. Collins, as always, and we'll continue to keep everybody updated as we progress. We'll now move on to our feature presentation today of a Grand Rounds. Before that, just want to mention, we of course have no Grand Rounds next week. I hope everybody has a wonderful and restful and thankful Thanksgiving. And the week after that, we don't have the title for it yet. It's Dr. Philip Sunshine. He is going to be our presenting as our Hewlett Award winner. As you may know, Hewlett Award is the largest award we give out in the Department of Medicine. And we don't always give it to just somebody within the Department of Medicine. Dr. Sunshine is a neonatologist, and he has been a neonatologist for over 50 years at Lucile Packard. And if you just look up, pretty amazing career. He's credited for saving over 30,000 vulnerable babies and training some of our most elite neonatologists in the country. So looking forward to hearing more from him and sharing more about his life and career in our next Grand Rounds, which will be in person as well. We'll also talk about a little bit more that day, but we have a Grand Rounds that'll be hosted by Dr. Mahoney the day after that, December 2nd. Just want to put that on your radar a little early, and we'll promote that again as it gets closer. And then we have Dr. Braunwald, Eugene Braunwald, who will be presenting remotely as well the week after that. So thanks so much, and thanks to Leah. And so today we have really excited to have presenting with us Doctors Catherine Blish and Dr. Tracy McLaughlin. I'll introduce Dr. McLaughlin first, who will be presenting first. I think many people, including myself, have been really excited to hear them speak ever since this article that they published a few weeks ago has been out there and in the news a lot. Dr. McLaughlin is a professor of medicine within our endocrine, gerontology, and metabolism division. She graduated from Stanford with honors, distinction in human biology, and MS in public health. She also at UC Berkeley and UC San Francisco. She completed her residency at Seneca Valley Medical Center and her fellowship here at Stanford. She's a member of BioX Diabetes Task Force Chair. She's a member of the Maternal Child Health Research Institute. And one thing I'll say, both for Drs. Blish and McLaughlin, I apologize. I won't go through all the honors and awards you both have just in essence of time, but there's a long list of stuff I got the opportunity to review. Dr. McLaughlin's research really focuses on between obesity and insulin resistance. She's done all sorts of different variations on that and various types of research throughout the years, including looking how weight loss studies and dietary restrictions relate to surgical interventions and metabolic changes, or weight gain studies in which controlled excess calorie is used to a variable degree to affect insulin resistance. Dr. McLaughlin, I remember when I was training, I still remember training many years ago, rotating with you and learning about medications like exenatide, and I learned a lot from you then, and I'm sure many people have learned a lot from you throughout the years. And so thank you for being with us. We also have Dr. Catherine Blish, who's also joined us on Grand Rounds before, and she's been a key member of helping keeping the community updated, particularly in regards to COVID, but that's just one of many aspects of her research career. She's also professor of medicine within infectious disease. She received her BS in biochemistry at UC California, Davis, as well as an MD PhD at immunology at the University of Washington. She's the Associate Dean for Basic and Translational Research here at Stanford and an investigator of the Chan Zuckerberg BioHub, also a member of BioX, the Maternal and Child Health Research Institute. She's the Associate Director of the Stanford Medical Science Training Program, and also was recently elected into the American Association of Physicians. For her lab, her goal is to develop new methods to prevent and control infections for a better understanding of immunology through clinical immunology, microbiological pathogenesis, and innate cell biology. And so this article they recently published was SARS-CoV-2 infects human adipose tissue and elicits an inflammatory response consistent with severe COVID-19. You may have heard of it if you didn't through our community, because it got a lot of attention really throughout the country. It's definitely been an interesting topic and helping us understand exactly why we've been so busy and why many of our patients with COVID, especially young ones, are oftentimes more obese, and helping us understand that's been really, really, I think, exciting for a lot of us to kind of give us an understanding of what we've been through and why. And so they're here to talk about the role of adipose tissue in COVID-19. Thank you, doctors, for being with us. I'll turn it over to you, Dr. McLaughlin, who'll be speaking first.
Great.
Thank you, Errol, for that very, very generous introduction. There we go. Okay, well, it's a great pleasure to speak about our research. This is definitely a great example of what I love doing at Stanford, which is team science. So this study came out of not only Catherine's and my labs, but also included cardiothoracic surgery, Jack Boyd, bariatric surgery, Dan Azaguri, and pathology, Gary Nolan in his lab. So we're going to talk about this topic that has sparked a lot of hypotheses, obesity and COVID-19 and the role of adipose tissue. These are our disclosures, and below is a link to the bioRxiv publication, still currently under review at Science and Translational Medicine. So I will go through first the epidemiology linking obesity to severe COVID-19, and then the potential mechanisms, and then Catherine will present our study results. Okay, hold on, let me get rid of one thing so I can see better. Okay, so obesity is now clearly established as a major risk factor for severe disease in COVID-19. So initially this was measured mostly as hospitalization and mechanical ventilation. And here you can see the early data from China and in Europe. On the left, you can see data from China showing the severe disease in the red and the non-severe disease in the blue in non-obese patients here on the left and obese patients on the right. And then this graph shows the linear relationship with BMI, starting even down, below a BMI of 20. And as the BMI increases, the probability of severe disease increased. And then this is a study from France in 124 people showing that in the bottom, you can see it clearly in the red box, that for a BMI of 35 or greater, the odds ratio of mechanical ventilation was about 7, both in univariate and multivariate analyses. Then why am I not advancing? For some reason, my slide is not advancing. There we go. And then the wave hit New York City, and we got similar data from the New York study, which was over 5,000 adults with laboratory confirmed SARS-CoV-2. And you can see down at the bottom, a BMI of greater than 40 was associated with about a 2-fold risk of hospital admission and of ICU admission. This study then was published. This is in Kaiser in Southern California, over 6,000 people. And what this study clearly shows is that particularly in individuals aged under 60, which is a trend shown in other studies as well, that particularly under 60 demonstrate increased risk from obesity, that a BMI of greater than 40 and greater than 45 conferred a 12 to 17-fold risk of severe disease from, this actually one is for death, from COVID-19. And here you can see there's also an increased risk in those over 60, but not quite as dramatic, a 3-fold increased risk. This study also showed something that pops up in some studies, but not all, which is an excess risk among males. Here you can see that among males, the relative risk is 4 to 10 for obesity, whereas in females, it's not even statistically significant. So that shows up in some studies. This is now a meta-analysis of 75 studies published on obesity and COVID-19. And these are what the relative risks are from this compilation of data. Even testing positive is increased among obese versus normal body weight at about 1.5 relative risk. Hospitalization is twofold increased. ICU admission about twofold increased in mortality, about 1.5-fold increased relative risk. And I want to show this. This looks busy, but let me just show you the main point of this slide. So this is severe COVID-19 and various outcomes by BMI split into a few different categories. This is all BMIs, all ages together, but I wanted to show you this J-shaped curve because you can see that hospitalization, ICU admission, and death all have a nadir of around 23 to 24 BMI, above which the risk increase. And this is important because some of the other studies I just showed you really focused on a BMI over 40. And when vaccines were coming out at the beginning, remember doctors are being very strict about what level of obesity allowed people to get a vaccine. Remember that? So it actually is any BMI above 24 is starting to increase the relative risk of bad outcomes. This is mechanical ventilation, which looks more like a linear association. And down here, you can see each of these different lines. You don't have to know which each one is, but these are all different age groups. And it's pretty much the same for each age group. Here, the risk goes up with BMI starting at around 23, 24. This is older individuals over 75, where ICU admission is more of a linear risk relationship. So we can move on to this slide. So there's been a lot of chatter about visceral fat. And these studies are popping out. They're small, but they do consistently indicate that it appears that visceral obesity may be a particular risk factor for severe COVID-19. So let me walk you through these. This is on, excuse me. These are studies where, everyone gets their CT scans of their lungs and they're able to go back in retrospectively and measure visceral fat, which is so smart. We should do that here. But this one is in 144 patients in Italy who had COVID-19 and pneumonia. And you can see that admission to the ICU was associated with a 30% higher visceral fat and 30% lower subcutaneous fat independent of age and sex. And that the ratio of visceral to subcutaneous fat, which is something that we use a lot because it's looking more at distribution of fat rather than total fat, right? So the ratio shows that you tend to be more viscerally obese. And this was associated with a BMI adjusted risk of ICU admission with an odds ratio of 20% increase for, an odds ratio for each 20% increase in the ratio of 1.25. And in this study, the area under the curve for receiver operating characteristic curve was 0.728, which is pretty good. This is a separate study showing that here we have waist to hip ratio and waist circumference, which are not as accurate as a CT scan, but these both perform better than BMI alone in predicting severe X-ray scores in patients with COVID-19. And these studies actually show another thing that's starting to emerge and has garnered great attention, which is, does epicardial fat, which is also present in viscerally obese individuals, in particular confer risk for severe COVID-19, because if there's inflammation in the fats or the fat is participating, if you have fat around your heart, maybe that's leading to bad outcomes. And there's a bunch of other data showing that the mass of fat around your heart is associated with poor cardiac outcomes in general. That's independent of COVID. And so this study in France showed that down here, the main risk factors for ICU admission or death were hypertension, greater lung involvement, and epicardial adipose tissue volume. And here they did a multiple regression analysis, and you can see that in all of these models, the increase in epicardial adipose tissue volume continue to predict poor outcomes in COVID-19. This study down here I included because it shows the same thing in younger individuals, and there is really a paucity of published studies in young adults and children. But then anecdotally, we keep hearing about obese kids and how important this is, and I really hope to see some publications soon. But this study showed this is from Wuhan, China, that fat in the liver and fat around the heart predict disease severity in young adults aged 18 to 40. Here you can see the moderate versus severe disease with a measure of liver fat, and down here with a measure of epicardial fat volume. Subcutaneous fat thickness was not a predictor of severe outcome in this study. So I would say that we're, there's definitely supportive evidence. These studies are small. I don't know if we'll get a big study, but it does appear that visceral fat, epicardial fat, liver fat are all associated with poor outcomes. And, it's really interesting. This is not the first virus that has been worse for obese individuals. In the H1N1 pandemic, a BMI over 40 had an odds ratio for death of 2.8, and over 45, an odds ratio of 4.2. And this wasn't fully investigated as to why. And that's what I think our study has done, and hopefully may even have implications for future viruses to come. But adipose tissue is a target for viruses. There have been other studies where, mostly in vitro or in animals, they infected various cells with viruses. So adipocytes supported infection with H1N1, and adenovirus, adipose stromal cells. You can kind of go through the list and see that multiple cells that populate adipose tissue, which, as you know, is not just adipose cells. There are a myriad of cells present in that tissue, which have been infected with other types of viruses. But again, these are mostly not human studies. These are mostly animal studies. Then comes the question, why is obesity a risk factor? And there are many hypotheses, all of which are very, very valid. But the first is that metabolic syndrome is present. The more obese you are, the more likely you are to have insulin resistance and all these metabolic abnormalities, which include hypertension, hypercoagulability, inflammation, endothelial dysfunction, latent CAD, and hyperglycemia sometimes. Another hypothesis is just, mechanics of breathing and respiration, where you've got a heavy, heavy chest wall. And then the third is an altered immune response. So, however, because ACE2 was, discovered to be present in adipose tissue, or thought to be present in adipose tissue, this hypothesis arose that SARS-CoV-2 actually binds H2, which, as you know, is the receptor for the spike protein, and may enter human fat cells. And this is a GTEx publication. When this first all came out about 2 years ago, there was a lot of there were a lot of hypothesis papers actually published, and they were based on this. So the GTEx studies show here that adipose, subcutaneous, and visceral express ACE2 in the tissue. These are not cells, adipose tissue, but both depose. And here you can see all these other tissues. And there also were some studies, with protein signatures. This is RNA, but there are protein signatures also showing ACE2 in adipose tissue. And the theory is very nice. SARS-CoV-2 spike protein binds to ACE2, becomes engulfed into the cell, which then not only allows viral entry, but downregulates ACE2. And ACE2, as you probably know, is anti-inflammatory. ACE2 promotes the metabolism of angiotensin II, which is pro-inflammatory, to angiotensin 1,7, and thereby theoretically could decrease all these actions of angiotensin II, which may contribute to acute lung injury adverse myocardial remodeling, vasoconstriction, and vascular permeability. So it was a very attractive hypothesis. And further, SARS-CoV-2, if it's getting into fat cells, may augment peri-organ fat inflammation and potentiate vital organ damage. And most of this interest was around the heart because, as you recall, at the beginning of this, there were all these cardiac events. 28% of people infected or severely infected were having, some cardiac event. And so if this is getting into the fat around the heart and replicating and causing inflammation, it could potentially lead to insulin resistance and inflammation and fibrosis and cardiac, either acute event or heart failure. So we hypothesized that SARS-CoV-2 infects cells in adipose tissue through ACE2 and causes an inflammatory response, in which adipose tissue may serve as a viral reservoir for prolonged replication, shedding, and seeding of adjacent organs, and also as a stimulus to inflammation in the adipose tissue, likely from direct pathogen insult. This could lead to systemic cytokine effects, including cytokine storm, inflammation, endothelial dysfunction, and hypercoagulability, all of which we see in severe COVID disease, and could contribute to regional inflammation in peri-organ fat depots, causing damage to adjacent organs such as the heart, intestine, or kidney. So just very recently, as we were submitting our paper, a very supportive paper came out in Cell Metabolism by James Lowe's group, showing that SARS-CoV-2 reaches adipose tissue in infected hamsters and induces a pro-inflammatory transcriptional response. And here you can see that in visceral adipose tissue and subcutaneous adipose tissue, under mock and SARS-CoV-2 infection. And these are not in the laboratory, which is what we did. These are actually infected hamsters, that you can see an increased PCR signal clearly in the visceral and the subcutaneous adipose tissue of these infected hamsters. And here is just a volcano plot showing in the visceral adipose tissue that relative to mock, there are a bunch of genes that are upregulated and downregulated in the infected animals. And when you take a deeper dive, you can see that the majority of these genes that are upregulated are related to defense to viral infection. And here you can see in particular, there are a multitude of chemokines that attract leukocytes to the site of infection. And so this was very, very in line with our hypothesis. And in fact, this group also isolated human adipocytes from breast tissue in 2 individuals and showed that in the lab, these human adipocytes could support SARS-CoV-2 infection. And here you can see the infected versus the mock conditions. So there are multiple questions that we have that we wanted to answer in our study. Number 1, does infection of adipose tissue occur in humans in vitro and during natural infection? Number 2, what cell types in adipose tissue are infected? Number 3, how is the virus entering adipose tissue and cells? And is it via ACE2 as hypothesized? And number 4, what are the inflammatory consequences of infection in human adipose tissue? So this is an overview of our study. We harvested, I will say Catherine was such a trooper with this. I couldn't find anyone to do this study. I wanted to just look at autopsy samples, and I think I either called or emailed Catherine, and within 24 hours, pretty much, we were harvesting tissue. Jack Boyd was getting us tissue from a cardiac patient. So we had harvested tissue from bariatric surgery patients, Dan Azaguri, and cardiothoracic surgery patients, Jack Boyd. And we would harvest the tissue, both subcutaneous and visceral in the bariatric, and epicardial and pericardial and sub-Q in the cardiac patients. And then we would run it to the lab and we would digest it when you digest adipose tissue, you can isolate different cell types. Basically, the collagenase digest the collagen, so the mature fat cells float up to the top, and the pellet stays in the bottom, which contains all the other types of cells, including the immune cells. Some of these cells we put in vitro in a dish and differentiated them. So this contains preadipocytes. The preadipocytes adhere to a plate, so it's easier to work with them, and we differentiated them and infected them on different days, as compared to mock conditions. And then we also infected the mature adipocytes. So most of our comparisons are comparing in the mature adipocytes and in the stromal vascular cells infected to mock conditions. And then we did a number of experiments, including PCR, secretome, flow cytometry, and single cell RNA sequencing that Catherine will review with you. And so that's my last slide. I'm going to turn it over to Catherine to share our results.
Okay, great. Thank you, Tracy, for really nicely outlining the premise and overview. And I just wanted to, Tracy already alluded to it. This is a picture of the, preprint that we have up if anyone wants to look at it. And I just wanted to call out in particular Giovanni Martinez-Colon, who did all the BSL-3 work to do this, which is no small feat, because working with fat tissue is hard anyway because it floats. And doing that in a spacesuit under BSL-3 conditions is a particular challenge. And Kalani Razzanieri, who did a lot of the bioinformatic analyses for the single cell data that I'm going to talk about. And of course, the whole village that it took to do this study, in which we really tried to take a deep dive into SARS-CoV-2 infection of adipose tissue and what we could learn. So the first question we really wanted to understand that wasn't addressed even in the study that came out around the same time that Tracy referred to is which cells were infected. And again, as she alluded to, within the adipose tissue, there are multiple cell fractions and many, many cell types. The mature adipocytes are the fat cells themselves, they float, but a number of cells pellet down if you spin this out. And those are the stromal vascular cells, which consist of a mix of preadipocytes, endothelial cells, and multiple immune cell subsets. And the prior study had really only looked at the mature adipocytes in those 2 breast tissue samples. And our first question was, is the stromal vascular cells, are those cells infected? And what we did was we took this tissue, as Tracy alluded to, collagenase digested it, and then took this pellet of cells, the more typical immune cells, and then did a number of different approaches to it. We took this into the BSL-3 and Giovanni infected it with SARS-CoV-2 or did mock infections. And we did quite a number of different assays to assess for whether there was true infection, including RT-PCR for SARS-CoV-2 genomes. We did some flow cytometry. And we also, and very importantly, treated these samples with remdesivir as well to try to determine if this was an actual active ongoing replication versus a more abortive infection. And with the flow, we could identify many of the different cell types within this population, including dendritic cells, macrophages, B and T cells, endothelial cells, and the preadipocytes. So what did we find? So first, this is what the tissue looks like. We took in parallel samples from the same patient in general that consisted of both the subcutaneous and the visceral adipose tissue. I'm going to refer to those as SAT and VAT for the rest of the presentation, and the SAT will always be in blue and the VAT in the kind of shades of brown, orange. And again, you can see here this kind of heterogeneous cell population. And the reality is if we infect this tissue, what we observe is robust infection. And this is a log 10 scale. So what we're observing is a 20 to 50-fold induction of SARS-CoV-2 reads in a sample that we exposed to the virus versus not in both the subcutaneous and the visceral adipose tissue. So really robust infection. Now, it remains possible that this is uptake of the RNA and not actual replication. To address this, what Giovanni did was he set up a really nice time course looking at infection where he exposed the subcutaneous, in this case, adipose tissue, to the virus for 2 hours, washed it off, and then let this culture go for up to 96 hours. And what we observe in this situation is an induction in a significant amount of SARS-CoV-2 reads. And importantly, this is significantly inhibited by remdesivir, a direct-acting antiviral. And this really suggests an ongoing replication because the remdesivir is an RNA-dependent RNA polymerase inhibitor. So what we're seeing is a difference between the ability of the virus to replicate. And we recapitulated this as well in the visceral adipose tissue, seeing again this expansion in reads. So as I alluded to, this is a heterogeneous cell population. And our next question was, which cell type is actually infected? And to address this, what Giovanni did was he did the infections in the same way and then did the flow cytometry panel that I mentioned. And what I'm showing here are the frequency of cells in which we're detecting now protein, intracellular SARS-CoV-2 N protein in the various cell subsets, dendritic cells, macrophages, B cells, T cells, endothelial cells, and preadipocytes. And what you can see is that consistently we're seeing the highest level of viral detection in the macrophages, potentially some in one of the subject in the dendritic cells, but by and large, it appears as though the macrophages are the primary target of infection in the SBC. And this was somewhat surprising, but is paralleled by data showing that we can detect SARS-CoV-2 reads in macrophages in BAL samples from infected patients, and also from data from the original SARS and MERS. Now, what was somewhat more surprising was that we really detected very minimal ACE2 expression in the SVC. If we looked by flow cytometry, we only detected ACE2 at an incredibly low level on 2% of the or so of the macrophages in the SAT, and not at all in the VAT. And we really didn't detect any transcript level. So this entry into macrophages appears to be not mediated by ACE2 entry. We next wanted to really further understand what was happening in this infection, better identify the target cells, and more importantly, do a simultaneous dive into what inflammatory pathways were activated in this setting. And therefore, we undertook a fairly extensive experiment to perform single-cell RNA sequencing to identify both targets and inflammatory pathways. And in this case, we again had 3 independent subjects from which we had matched visceral and subcutaneous adipose tissue. We were able to obtain this stromal vascular cell compartment and do mock versus SARS-CoV-2 infection. We tested at a 72-hour time point, and we ran this through a 10x single-cell RNA sequencing platform and then sequenced it. And what we identified was a remarkable 23 different unique subsets of cells based on this transcriptional profiling. And I won't bore everyone with taking them through each of these, but you can see some major cell subsets. Up here are those macrophages, and they actually form 2 unique transcriptional subsets, which we'll refer to as C2 and C12 for cluster 2 and 12. But we also see other subsets of cells like, B cells, dendritic cells. Down here are all the preadipocytes, and we have endothelial cells and so forth. So we can identify many unique cell types, and this leads to many questions. First, where is the infection? And second, how does infection actually change these depots? So if we take that same projection where each of these dots is an individual cell, and each of the dots are closer in space, 2 dots that are more transcriptionally similar, and we color it now, instead of by cluster, by the condition, where the light blue are the subcutaneous adipose tissue, the dark blue are the subcutaneous adipose tissue that we've infected with SARS, and then we've got the same thing for the visceral and the adipose tissue in the infected and uninfected condition. And what we see is that there's actually a lot of overlap between the light and the dark blue and the light and the dark brown, indicating that we're not seeing a massive transcriptional shift in the effects of infection. Now, interestingly, we do see that visceral and subcutaneous preadipocytes down here are quite transcriptionally unique and have many unique features, which is heralded by what we've already learned, that visceral fat is quite unique and is actually uniquely predictive of SARS-CoV-2 bad outcomes. So now if we look at this, we have the ability with this single cell data to dive into each individual cell and say which cell is actually infected. And specifically, which cell has SARS-CoV-2 reads. And if we now color this same plot by SARS-CoV-2 reads, you can see that really this one subset of macrophages is harboring most of the infection. Almost every cell, this is counts per million of SARS-CoV-2 reads. And you can see that almost every infected cell is here. There's some very low level in other tissues. To look at this another way, if we look, this cluster that I'm referring to is actually a very inflammatory cluster of macrophages. Even in the mock condition, this cluster's most unique characteristic is high expression of two pro-inflammatory transcripts, IL-1 beta and CCL3. And you can see that 85% of the SARS-CoV-2 reads are found within this cluster. And in fact, at a very high level, about 4,000 reads per cell, so a very impressive level. We do detect about 11% of the reads within the other more conventional macrophage subset that's characterized by expression of lysozyme and also an alarmin, S100A8, and really very minimal reads and at a very low level in any other cell type. So this leads to, again, further questions about what are the characteristics of these macrophages? Why does the virus get into them? And how is it entering? And so if we just focus in on these two macrophage clusters, so again, we have cluster C2, the infected cluster, we're looking at the SARS-CoV-2 reads, cluster C12, the slightly less infected cluster. If we look, these are both clearly macrophage clusters. CD68 is a canonical macrophage marker. But if we now look for ACE2 expression, you can see a distinct absence of any purple dots. We're actually not detecting a single ACE2 read in these cells. And what I'm showing here on the right is an analysis that Kalani put together, looking at expression of many genes that are associated with SARS-CoV-2 infection, including putative entry receptors. And you can see that, and this is colored by their average expression, here's ACE2. And so whether we look in the cluster 12 that's relatively uninfected, or the infected sample, or the infected cluster from the other cluster, there's really just no ACE2 expression. But interestingly, there are several other transcripts for receptors that have been putative alternative entry receptors, including DSG, NRP1, and NRP2. And what you can see is that particularly in the infected sample, we do see upregulation of transcripts for these receptors in the infected sample, suggesting that maybe one or more of these could help to mediate viral entry. Now, we also see broad induction of many inflammatory genes and genes associated with processing of the virus, including cathepsins and a number of interferon-stimulated genes. You can see IFID-M1, IFID-M2, and interferon receptor here. So what we're observing is that these macrophages are infected. It is probably not through ACE2. It might be through an alternative entry receptor, and more work needs to be done to detect that. And we're seeing a robust inflammatory response in these macrophages. Now, if we look in more detail at this macrophage cluster, and in this case, we're trying to investigate what is it about this macrophage cluster that allows it to become infected. So in this case, what I'm looking at is a heat map, a volcano plot showing genes that were upregulated in this cluster C2, the cluster that stores more robust infection compared to the relatively uninfected cluster. And what we see is that already, this is in the mock condition, these cells have never seen virus. This cluster is expressing high levels of chemokines like CCL8, CCL4. It's expressing high levels of the cathepsin involved in viral processing. You can see IL-1 beta here and Marco, whereas the relatively uninfected cluster is expressing high levels of lysozyme, a canonical marker, although some inflammatory markers too, including alarmins. And importantly, if we then did the same comparison in the infected, comparing the infected to the sample in C2 to C12, we see just a further polarization of this phenotype. We don't see a lot of unique genes, which is why they probably didn't differ that much in the transcriptional space. But instead, what we observe is a further upregulation of these already inflamed genes. So the next thing we wanted to do was to make sure that this is really leading to inflammation in a way that causes cell-cell communication, because transcriptional profiling is very important, but this is purely RNA, what I've shown you. And we're curious if these inflammatory proteins are also being produced. So what Giovanni next did was he set up another infection and actually assessed cytokine levels in the supernatant of these infected cultures. And this is, I apologize, a very busy table, but I'm just going to highlight a few things. So for instance, if we start at the top, what we see is this IP10 chemokine, which is one of the most robust antiviral chemokines that's induced in an interferon responsive method early in infection. is actually 20-fold induced in the subcutaneous adipose tissue, almost following infection, and is 17-fold in the visceral adipose tissue. And this is really intriguing because if we look at its known role in COVID-19, we and others have characterized that this is elevated in acute cases and in critically ill patients. It's associated with disease severity and ICU admission, and it is a predictor of disease progression. And we can continue looking down this chart and find a number of other similar cytokines and chemokines. PGDF alpha alpha, also associated with severe disease and ICU admission, is about 5-fold upregulated. VEGF is similarly associated with severe disease and is elevated and is thought to have a role in COVID-19 brain inflammation. It's also 2 to 4-fold induced, and MIF, an inflammatory chemokine and cytokine, also elevated in inflammatory patients, is also about 10-fold induced in these samples. So what we're seeing is that this infection of the stromal vascular cells is capable of inducing these cytokines that are strongly associated with poor outcomes in COVID-19 patients. And this also has an interesting additional effect, because if we now look at the preadipocytes, I mentioned that in the stromal vascular cells, there are preadipocytes which can pellet, which will then eventually become preadipocytes. And these are not highly infected. But what we can see is that there are a number of unique transcriptional subsets of these cells. And they're not massively changed if you just look at the global UMAP projection by infection, but they're quite distinct between the visceral and the subcutaneous tissue, as I alluded to earlier. But what is interesting is these preadipocytes are inducing a massive inflammatory response in response to the inflammation induced by the infected macrophages next to them. So if we look at IFI-27 and IL-6 as markers of inflammation, we can see massive expression of these transcripts. And if we look at this in more detail, what we found was somewhat surprising, was that actually the subcutaneous adipose tissue is responding more robustly to infection than the visceral adipose tissue. So what I'm showing here is an overall cytokine storm for induction of pro-inflammatory cytokines in the subcutaneous and visceral adipose tissue in the mock versus the infected culture. And what you can see is that there's a greater induction in the subcutaneous adipose tissue, but this may be in large part because the visceral fat is already more inflamed. So you can see that the visceral adipose tissue is starting at a cytokine level that's just as high as the infected sample. And it's not increasing significantly because it's already potentially at its maximum level. If we look at this, instead of looking at cytokines, at the interferon stimulated genes, which are an important antiviral protein, we again see a greater induction in the subcutaneous adipose tissue, but we also see some induction in the visceral adipose tissue in this case. So all this I've told you about so far has been just in the stromal vascular compartment. And so we next wanted to investigate what was happening in the mature adipocytes, those mature cells that are floating in the sample. And these, as I alluded to, are much more challenging to work with because it's harder to physically wash the virus off them and so forth. Nonetheless, Giovanni did really yeoman's work to look at infection in mature adipocytes from subcutaneous and visceral adipose tissue. And what I'm showing is a fold change in the infected relative to the mock. So again, massive infection of the subcutaneous and visceral adipose tissue on the order of 10,000 copies. And in this case, I'm showing both the genomic and subgenomic RNA quantification in order to try to get at the idea, because subgenomic RNA should really only be produced upon viral replication to really prove this as viral replication. To look at this another way, given the difficulty in making sure that we aren't re-amplifying our input virus, we also differentiated adipocytes from preadipocytes. And if we did this, as I already alluded to, the preadipocytes we found in our single cell data were not infected. And in fact, we find that again here on day 0 before they've differentiated. We don't see any SARS-CoV-2 reads. But as these cells march through their differentiation, we begin to detect their ability to be differentiated. In addition, if we repeat that remdesivir experiment, we observe that we can see, again, evidence for spreading infection in these differentiating mature adipocytes, which are in the process of differentiating. And this is, in fact, inhibited by remdesivir, the antiviral. So finally, this really leads to the big question of, does this matter in vivo? And in this situation, as Tracy alluded to, we were fortunate to collaborate with Gary Nolan's lab and also some colleagues in Germany, Matthias Matter, Christian Schurch, and Xizun Jiang, postdoc in Gary's lab, who helped us identify some adipose tissue from COVID-19 deceased individuals. And this was actually somewhat more challenging than one might have thought. I contacted many, many groups. And in general, the fat tissue is sort of lumped in with the other tissue. But this one group was able to find some. And importantly, in 8 autopsy samples that we looked at, we detected SARS-CoV-2 reads. And you can see the cycle threshold values where anything less than 37 is considered significant here in the fat. And it is much lower than in the matched lung sample, as we might expect, but it is detectable. And in fact, a lower cycle threshold reflects a higher level of virus. And you can see that, in fact, the levels in the fat are higher than that in the adjacent tissues. So for instance, if you compare the cycle threshold in the fat here compared to the heart, it's reflective of higher viral load. Interestingly enough, this first autopsy sample is somebody who passed away many weeks following their initial infection, and we were unable to detect any SARS-CoV-2 reads either in the lung or any other tissues, which helps to show specificity. Now, Xizun was able to also perform some immunohistochemistry on an epicardial fat sample in this in one of these patients. And what we're looking at here is this epicardial fat. I'm showing a blow up of right here. You can see the myocardium and the epicardial fat. And if you look here, what we're detecting are SARS-CoV-2 RNA by RNA in situ hybridization in this adipocyte. And what we're seeing here is an inflamed mononuclear cell. If we look at another tissue section, here we're again looking at the epicardial fat. We again see detection of SARS-CoV-2 surrounded by a dense mononuclear cell infiltrate with many mononuclear cells. And if we look at that myocardial sample, again, what we can see is really inflamed fat with a periodic detection of SARS reads and mostly a really strongly induced inflamed fat. So if we bring this to in conclusion, what we really find is that SARS-CoV-2 infection of fat can lead to infection of both mature adipocytes and macrophages, which induces a broad inflammatory response. And this occurs from multiple depots. And the infection drives this inflammatory response that includes secretion of molecules associated with severe COVID, and that there are these 2 cellular targets of infection. And that we can detect this in adipose tissue associated with an inflammatory infiltrate in SARS-CoV-2 infected patients. And so this really leads to the overall conclusion that SARS-CoV-2 infection in adipose tissue may contribute to severe COVID-19 in obese individuals via expanded viral reservoir inflammation in periorgan fat depots. But there are, of course, many more things we need to understand, including the mechanism of entry, which will be necessary to determine whether there are specific blockers for this, whether lean fat responds similarly, whether metabolically unhealthy individuals respond with greater inflammation, and whether infection of adipose tissue plays a role in long COVID or post-acute sequelae of COVID, also known as PASC. And so with that, I tried to highlight the major players as we went, but I just want to thank our funding from the IMA, the American Diabetes Association, the NIH, both NIAID and the NIDDK. And particularly, I highlighted Giovanni and Kalani, who were lead authors on this paper, others from my lab who provided support, our collaborators, and of course, Tracy's lab, our ability to recruit the patients, our surgical colleagues, and others who helped with this study. So thanks very much, and we'd be happy to take questions.
Thank you so much, Drs. Blish and McLaughlin. That was really amazing, and we learned a lot about the pathogenesis and the connections. We are hitting right at 9 o'clock, but this is one we were expecting to get lots of questions, and we already, of course, do. We, of course, won't get to most of them, but if it's okay to hang back for a few minutes, try to get to a few, that'd be great. And I'll, of course, just go with some of the most voted up ones. So please do vote up ones that we're likely to get to. Dr. Leibowitz asks, is there a difference between visceral and subcutaneous fat as a surrogate for male versus female sex?
I can address that. Males have a lot more visceral fat, particularly as body weight goes up. And if you just look at abdominal fat, up to 50% of the abdominal fat in males can be visceral. So I don't know about a surrogate, but males will be more prone. They have more visceral fat. So if visceral fat's important, it may selectively impact males, if that's, I think, where he's going with the question.
Great. I'll keep trying to fly through questions. Dr. Stefanik asks, can you remind us how many male versus female subjects you had in the study?
Yes, hold on.
Do you have a slide for that? Yeah,
I do.
The majority of our patients are female, just from bariatric, but Catherine's probably pulling up the exact number.
I'm pulling it up. Actually, the majority, well, so for the autopsy, the majority were male. It was 6 male and for total samples and 3 female. And I should remember that. I believe it was, oh man, I dropped it out of this deck, so I don't have it handy.
The cardiothoracic patients, one thing to really make sure everybody understands is that the power of having multiple depots. So, I mean, we had mostly probably females from the bariatric, and I think the cardiothoracic was a male, but we had a visceral fat, sub-Q fat, pericardial fat, epicardial fat, and the autopsy showed the same thing. It was multiple depots. So this is really important because it's a broad effect in adipose tissue. And it does appear that it can infect cells and people of both sexes.
Great. Dr. Kleber.
By the way, I just found it. It was roughly 65% female in the samples that we did for the in vitro studies.
Great, thank you. Dr. Kleber asks, is there association between the amount of adipose tissue and long COVID? Thank you.
Catherine, do you have that? I can actually address that if you.
I'm sorry, the question I was looking at.
Sure. Association with adipose tissue and long COVID.
I'll go. I looked that up last night to see it because there had been nothing published. And finally, there's one paper that just came out, but it's there's very little data on this. The one paper that was out was in healthcare workers in Italy who were exposed and who got COVID. And there was what appeared to be some association between increased BMI and long COVID, but it's just, again, one study. So that definitely is a very important issue that I think in the next, several months to years will require investigation. We are in fact looking at that in our cohort, and I also in fact looked at that data last night, but it's too preliminary to present at this point.
Well, and I believe Dr. Nadeau has done a deep dive into our biobank, and it did not, and Dr. Pinsky is aware of this data as well. He's one of the lead authors. My recollection is it wasn't a major factor that came out in our long COVID within our own biobank of roughly 500 folks so far.
Yeah, the way we need to really look at that too is, I think what we're trying to do right now is look at whether, we don't really have a measure of visceral obesity versus non-visceral obesity. And we also need to look at different age groups, because remember, this under 60 is what popped out in a lot of studies. So we want to look at the selective impact at those under 60. And what we're probably going to do is use a surrogate marker for insulin resistance and see if in the insulin resistance subgroup, there's an impact on long COVID with regard to BMI. So there's much more to do, and we'll probably be working on that over the next several months.
As we're coming up, I might just do one more question, and then we'll probably put a hard stop at 9:05, if that's okay with you. Dr. Chu asks, can you test if imatinib and bevacizumab suppress cytokine response in vitro?
Yes, we can. It's just an issue of logistics and doing it. I think I would argue that an in vivo model might actually be a better place to test something like that, where going back to that hamster data that from the cell metabolism paper that Tracy presented, where we might want to see a more systemic effect of a drug like that, but we could certainly see if it would block it in our in vitro cultures. I'm just not sure what ultimately that would tell us about treatment. But I'd be very intrigued to see if that would reduce the inflammatory signatures in the adipose tissue itself.
Great. Thank Dr. Siegel and Sprinkle. Sorry, it's 9:05 now. We should probably wind down so people get to other meetings and so on. But Dr. Blish and McLaughlin, thank you so much for that ton of information, valuable information, and really enlightening for us. So thank you for the work that you do and for sharing it with us today. I hope everybody has a great rest of the week and stay safe.